Good afternoon, ma'am.
Yeah.
Good afternoon.
So we will continue with
Afternoon,
the,
ma'am.
okay. So today we will continue with the structural organization of proteins, wherein already you know about the primary structure, secondary structure, tertiary structure, right? So today let us discuss about the quaternary structure of a protein. Okay. For this we are taking an example of hemoglobin. So hemoglobin's quaternary structure we are going to discuss now. So what is this quaternary structure referred to as? See, proteins when you say they may contain a single polypeptide chain or it can be more than one polypeptide chain. So just like insulin, as you know, it has two different polypeptide chains. The two different polypeptide chains, one we call it as A chain and the other one is B chain. So both the chains are held together via disulfide linkage, right? So like that, a protein can have not just a single polypeptide chain but more than one polypeptide chain. So proteins containing more than one polypeptide chain, they exhibit this additional level of structural organization. Okay, so this is what is quaternary structure, or in other words, quaternary structure describes the way in which the subunits are arranged together. See, if there are more than one polypeptide chains, the arrangement between the first unit and the second unit, okay, or if there are three polypeptide chains, the linkage between the first with the second, then first with the third, or second chain with the first chain, or the second chain with the third chain. So like that, each of the chain, how it links with the other polypeptide chains. Okay, that is what is the quaternary structure. So let's see again. Quaternary structure describes the way in which the subunits are arranged together and the nature of their contacts. I hope it is clear. So quaternary structure refers to those polypeptide chain which contain more than one polypeptide chains and their interaction with each other. Fine. So these subunits, they associate or they link together via the non-covalent interactions similar to those involved in tertiary structure. So while discussing the tertiary structure, you know, the myoglobin structure we were discussing, it has ionic bond also, hydrophobic interactions or hydrogen bond, right? Or van der Waals interactions. So like that, here each of the polypeptide chain can have all these non-covalent interactions, and again those subunits, the different subunits, they also are associated via the non-covalent interactions. Okay, so this is what is the structure of a hemoglobin. So hemoglobin, as you know, it is present in the red blood cells of the RBC. So this one single RBC or each RBC contains around 640 million molecules of hemoglobin. So the number of proteins, the hemoglobin proteins present in a single RBC cell is around 640 million molecules. Okay, so then you can imagine the number of the hemoglobin molecules present in our body, right? So you have to count with the number of total number of RBC cells, then each RBC containing around 640 million, then what is the total number of RBC might contain, right? Okay, now looking at the structure of it, so the RBC which contains the hemoglobin, this is a structure of a single hemoglobin protein. So you can see with different colors it is depicted. So one with the orange, green, purple and pink, right? So each one of this is a polypeptide chain, which means to say this hemoglobin has four different polypeptide chains. Okay, and out of this four, two are similar in nature, are similar in structure. So like this and this orange and purple. So the four different colors is given just to make you understand a hemoglobin consists of four different subunits, or the four different polypeptide chains. Now here, all the four chains are not different, but here it is only the two chains are different and the other two are similar. See, two alpha chains and two beta chains. Okay, so hemoglobin consists of four different polypeptide chains, out of which two are each are similar, but two are alpha chains and the two are beta chains. And each of these subunits or the polypeptide chain has a heme group in it. Here you can see the heme group, so that it has an iron, and that iron is the one which binds to the oxygen. So now you can understand each of the hemoglobin molecule, because it has four different polypeptide chains and a four heme group present in it and a four iron atom. Thus a single hemoglobin molecule is capable of binding to four oxygen molecules. Okay, so two alpha chain and the two beta chains, totally four polypeptide chains are present in a single hemoglobin. Fine. So quaternary structure, now you can understand, it is a larger assembly of several proteins. It's a larger assembly of several proteins. Sorry. Usually called as subunits. So as I was referring to all this time, so those polypeptide chains present are referred to as the subunits. So the subunits can be identical or they can be different, right? Just in hemoglobin, so there are two identical and other two are different, you can say. So it has two polypeptide, I mean two different chains like alpha and beta, two alpha and two beta, totally four subunits it has. When they are different, so what is the purpose of having a different polypeptide chains, which means to say each subunit tends to have a different function. So each of the subunit or each of the polypeptide chain has a function there in the protein. That is why it has so many number of proteins present in it, or a polypeptide chain. Now we have a common shorthand form to represent this, that is the subunits are here referred to as alpha are beta, gamma. So the Greek letters are used, where the alpha 2 refers to, this is a quaternary structure of a protein where it has two alpha chain or one single beta chain or one gamma chain. Okay, so that is how it is. A protein with, now protein, quaternary structure of protein in a hig baritive ಅಂದ್ರೆ ಅರ್ಥ ಏನು? So which means 2, 3, it has four different polypeptide chains or four subunits, in which when you write alpha 2, it has two alpha, one single beta and one gamma chain. Okay, that is how it is. Then hemoglobin, as you know, it is an iron containing oxygen transport metalloprotein present in the red blood cells of all vertebrates. So almost all the vertebrates, they have this protein hemoglobin, where it is an iron containing oxygen transport metalloprotein. Why metalloprotein? Because it has iron in it, right? That is why it belongs to the group of protein called metalloprotein and is present in the red blood cells. So it was in 1959, a British molecular biologist, that is Max Perutz. He was the one who determined the molecular structure of hemoglobin by using X-ray crystallography. X-ray crystallography is a technique which is used to find out the molecular structure. So it was Max Perutz who found out the molecular structure of hemoglobin, that was in the hemoglobin of a horse. A horse hemoglobin he had taken for his experiments, and wherein he found out the molecular structure that it contains these many amino acids and these are the number of polypeptide chains present and these many are similar and this is what is different. So all these details. So that is what he has found out. It was Max Perutz. He gets the credit of determining the molecular structure of hemoglobin in the horse hemoglobin. So it contains 574 residues because it is a vertebrate RBCs or the vertebrate hemoglobin. They are all similar in structure and function. So the same thing we can continue here. That is 574 residues. So residues, you know, it refers to the amino acids. So the hemoglobin consists of 574 amino acid residues, and for which of course he was awarded the Nobel Prize also. So in mammals, if you look at 97% of the RBC's dry content contains this hemoglobin protein. So around 97% of the protein present in the RBC is nothing but hemoglobin. And as I told you, each red cell, each RBC has around 640 million molecules of hemoglobin present in it. And you know what is the oxygen binding capacity? That is 1.34 ml of oxygen is bound to 1 gram of hemoglobin, which means 1 gram of hemoglobin can bind to around 1.34 ml of oxygen. And the structure, when you look at the diameter of this hemoglobin structure has 5.5 nanometer. That is in terms of nanometer we say. So here again you can see the four different polypeptide chains, and majority you can see it is alpha helix only. When all these spiral things, what you can see, it is alpha helix, and of course there are some random coils which are present. So random coils are nothing but those amino acid sequence which are not capable of forming the alpha helical structure there. Okay. So one hemoglobin can bind to four oxygen molecule. Why? Because each of these polypeptide chain has a heme group, and each of the heme has one iron. So four subunits, so four iron atoms, so four oxygen molecules can be bound to one hemoglobin. Okay. Then okay, the time that is taken for binding the oxygen by this hemoglobin, or we call that as oxygenation. So the duration of oxygenation is less than 0.01 second. Okay, so you know 1 second, right? How fast it moves, and here it is not even 1 second. It is 0.01 second. Again, it is not just 0.01 second. Less than 0.01 second is required for the oxygenation, which means to say so much of affinity it has got to bind to the hemoglobin or the heme. Now, the heme and the globin, you know, hemoglobin is it has a prosthetic group, that is the heme. Heme is a prosthetic group and globin is the protein part. Now here, the heme and this globin, they are produced at two different sites in the cells. So, you know where the heme is produced? The heme is produced in the mitochondria, whereas the globin is a protein, and you know that proteins are synthesized in the ribosomes. So protein, the globin part is synthesized in the polyribosomes, whereas the heme part, the prosthetic group of this protein, is synthesized in the mitochondria. That is, at two different sites they are produced. So you know about the structure of the heme, like as we were discussing about the structure of the myoglobin, I have told you. So this is a protoporphyrin, wherein this iron atom is bonded to, I mean, it has six coordination bond. So out of that six, the four bonds to the nitrogen of the pyrrole ring. See, porphyrin has four pyrrole rings in it. Each one of has a nitrogen. So the iron binds to the nitrogen of this pyrrole, then another one, two coordinate bond. This is the third one, and this is the fourth one, right? So it has four coordinate bond that is formed now. Then fifth coordinate bond is with the globin, that is with the protein part. The protein binds to this heme part, the prosthetic group. And what about the sixth coordinate bond? That sixth is the one that bind to the oxygen. Is that clear? So iron has six coordinate bond, out of which the four is with the nitrogen of the pyrrole ring, and the fifth is with the protein part, and the sixth is with the oxygen atom. So that is how the globin and this porphyrin, that is, they are bound together or linked together. Okay, this is an alpha chain, and similarly there are two similar polypeptide chains referred to as alpha, and two other similar polypeptide chains in the subunit referred to as beta, and each of the polypeptide has a heme group and has an oxygen. So heme binds to the polypeptide chain via an amino acid. The same structure what we have discussed for the myoglobin, the same holds good here also. So those who are not understanding the myoglobin structure can now at least you can have a look, or now we can try to understand. Okay. So let us see. Hemoglobin consists of, you know that it consists of two alpha chain, isn't it? And these two alpha chains, each one has 141 amino acid. See, one polypeptide chain, this is alpha, has 141 amino acid. Similarly, this also has 141 amino acids. So each of the alpha chain has 141 amino acids. Then coming to the beta chain, the beta chain or the beta subunit has each has 146 amino acid residues. So alpha is with 141 amino acids and the beta is with 146 amino acids. Now what is the total number of amino acid there? You have to count it as 141 into 2, then 146 into 2, then add both of them, right? That is the number of amino acids present in the hemoglobin, because hemoglobin contains all these 4 polypeptide chains. Okay. Now, together we call this as a tetramer. Tetramer because it has 4 different polypeptide chains, right? We write it as the designation given to this is alpha 2 beta 2. What does this alpha 2 represents? It has 2 alpha chains. And what is this beta 2? It has 2 beta chains. Okay. The beta chains, alpha chains, and each one with an iron atom, each one with an heme, and the heme has an iron atom, and that iron can bind to oxygen. So one hemoglobin can bind to 4 oxygen atom. Okay. Now, coming to the structure again, each of these 4 chain have a characteristic tertiary structure. So here, when you say quaternary structure, so each of those polypeptide chain or the subunit present, this is protein, they have to have a characteristic tertiary structure. So alpha chain has its own characteristic tertiary structure, which means to say it has its own folding. The overall folding is nothing but the tertiary structure, right? Then another alpha chain that also has to have a tertiary structure. Then beta with a tertiary structure, another beta with another tertiary structure. Now all these 4 together, the linkage is the one that referred to as the quaternary structure. The interaction between the subunits. Is that clear? When you say quaternary structure, it is how exactly the subunits are interacting with each other. Then they have to have a characteristic tertiary structure. So like myoglobin, if you have seen the structure, I have told you, it contains several helix joined by bends or the random coil. They don't have the beta sheet is not present here in both myoglobin and the hemoglobin. The secondary structure what is present is the alpha helix. Now, each subunit, it has a heme group. Then the proteins with the subunits, the proteins with the subunits are called as oligomeric proteins. So, what is oligomeric protein? So, oligo here refers to many, right? So, this protein has many subunits or many polypeptide chains. Hence, it is called as oligomeric protein. Now, if those subunits are different, then it is called heterooligomer. Hetero refers to it is different. Oligomer because it has more than one polypeptide chain. Now here hemoglobin because it has 2 types of subunits, an alpha and a beta, right? Hence it is called as heterooligomer. Now if a oligomeric protein, a protein with many subunits, if those subunits are identical, then we call it as homooligomer. So totally oligomeric proteins are nothing but those proteins with subunits. If it is similar, if the subunits are similar or identical, then it is homooligomer. If the subunits are excuse me, if the subunits are different, then it is called heterooligomer. Okay. Now, there is an extensive interaction that can be seen with these unlike subunits. So, which are the unlike subunits? Alpha and beta. So, interaction between alpha and beta is an extensive, where each alpha chain is in contact with both the beta chains. Like here, alpha 1 and alpha 2, right? Now, alpha 1 interacts with beta 1, alpha 1 interacts with beta 2 also. Similarly, alpha 2, that is the second alpha chain, that also interacts with beta 1 and alpha 2 interacts with the beta 2. That is, each of the alpha chain is in contact with both the beta chains. Okay. Okay, so far did you understand about the hemoglobin structure? Uh, One of you can unmute yourself and tell me. Is there any doubt so far? Do you have to
No,
repeat?
ma'am.
No, you're following it, right? Okay, right. So let's go with this alpha chain. So you know any polypeptide chain will have one end will be a free amino group and the other end will be a free carboxylic group, right? So the amino group, the free amino terminal is also called as N terminal and the free carboxy terminal is also called as C terminal. Now in the alpha chain or this polypeptide chain, N terminal end has valine. Valine is the amino acid present in the N-terminal end of the alpha chain, whereas in beta chain also the N-terminal end has valine. And coming to the C-terminal end at the carboxy terminal, the polypeptide chain which has a free carboxyl group, the end one or the terminal one which is not forming, which is not able to form a peptide bond with the next amino acid because there is no next amino acid, that is the last one, right? So COOH will be free there. So that is called a C-terminal amino acid. Now, which is a C-terminal amino acid in alpha chain? In alpha chain, it is arginine, whereas in beta chain, it is histidine. Okay, so it both in N-terminal alpha and in beta, it is valine, whereas C-terminal, it differs. In alpha, it is arginine, and in beta, it is histidine. The alpha and beta, they are held together by ionic and hydrogen bonds. So that is what I was telling. The unlike subunits or those polypeptide chains, they have an extensive bond between them at the linkage. And which is that linkage? It is both ionic and a hydrogen bond. Ionic interaction or the electrostatic interaction. Then they also have additional ionic bonds or the hydrogen bonds along with the hydrophobic interactions. Okay, so these are the forces which stabilize the quaternary structure. So one polypeptide chain with another polypeptide chain, it is ionic interaction, hydrogen bond and hydrophobic interactions will be there which held both those chains together. Now, coming to the type of hemoglobin, we have hemoglobin A, which is the major type of hemoglobin. And this is what we are discussing. Why major type? Because the percentage, the normal percentage in vertebrae varies up to 98% it is. So from 96 to 98% of the hemoglobin is of this type, hemoglobin A. What is hemoglobin A type? Which has alpha 2 and beta 2 chain. Whereas hemoglobin A2 is a minor form of hemoglobin which can be up to 1 to 3%, where it is alpha 2 and delta 2. Unlike the beta chain, here it has delta chain. So beta and delta, they differ in the structure. Okay, and here it is the delta 2. Now coming to the hemoglobin F type, or we call it as fetal hemoglobin. This both A and A2 are the adult hemoglobin forms, whereas F is the fetal hemoglobin, which means in fetus, the structure or the composition of the hemoglobin slightly varies, wherein you can see it is alpha 2 and gamma 2. Here it is not either beta or delta, but the chain what we have designated is the gamma. There again the composition of the amino acids is different from the beta and the delta. And this percentage is very less until they become adult, some 0.5 to 0.8% only. Okay. Otherwise, the normal or the major form what is found in all the vertebrate is hemoglobin A only. Okay, you have hemoglobin A as the major form, then A2 also is present, then fetal hemoglobin is also present, wherein it has, so alpha is common in all, whereas the beta chain varies. In A form it is beta, in A2 it is delta, and in F form it is gamma. Yeah. Now again, let us go back to the structure of what we were discussing. The interior, as you know, because it is present in the aqueous solution. Which aqueous solution? That is inside the blood, right? It's nothing but the water and all the cells. So the interior consists of the hydrophobic or non-polar amino acids. So any protein which is soluble will fold it in such a way that the outside will have hydrophilic amino acid and inside the protein will be hydrophobic amino acids. Why hydrophilic outside? Because it has to interact with that aqueous environment. Okay. So the non-polar or the hydrophobic amino acids are present inside, whereas the hydrophilic will be present outside the protein structure. The hydrophobic R group of helix E and F, they form the sites. Here also, just like the myoglobin, there are number, each of these subunit has A to H, that is the helix, wherein the helix E to F, E and F, they have a pocket-like structure into which the heme is present. This what is the same structure what we have saw in the myoglobin structure also. A to H is the helix in which E and F, between the E helix and the F helix is the heme that is present. Okay. Then inside again there are O2 polar amino acid that is present. So just now I said inside it is non-polar amino acids, but there are two polar amino acids present there. Now why they are present inside being polar? They're supposed to be outside, right? But these two polar amino acids, they have some functions to perform. Which are they? They are the two histidine molecules. They have a role in binding of heme to the oxygen. See the iron atom, okay, these things I told you, it has 6 coordination numbers, 4 with the nitrogen of the pyrrole atom, 5th with the globin, and 6th with the oxygen, isn't it? Now, histidine 93 is the amino acid known as proximal histidine. That is the only connection of heme group with the protein. So the 6th bond of this iron atom, the 6th coordinate bond of the iron atom with the globin is via this histidine 93. So the histidine present at the 93rd position of the polypeptide chain called as proximal histidine. So the iron atom binds to the globin chain via this histidine. So globin nalliruva histidine hemaliರುವ iron jothe link ಆಗುತ್ತೆ. That is how the positive group and the protein are held together, making it as hemoglobin. Okay, heme is not bound to the entire protein. The only connection yadra ಜೊತೆಗೆ ಅಂತಂದ್ರೆ it is with the nitrogen, it is with the heme group and the histidine 93. Okay, now don't scribble on your screen. ನೀವಿನ್ನು ಸಣ್ಣ ಮಕ್ಕಳೇನ್ರಪ್ಪ, ಕೇಳಿಸ್ಕೊಳ್ಳಿ ಸಾಕು. ಯು ಡೋಂಟ್ ಹ್ಯಾವ್ ಟು ಆನೋಟೇಟ್ ಎನಿಥಿಂಗ್ ಓವರ್ ದ ಸ್ಕ್ರೀನ್. ಬೇರೆಯವ್ರಿಗೆ ಡಿಸ್ಟರ್ಬ್ ಮಾಡಬಾರ್ದಲ್ಲ. ಓಕೆ, ನೌ where is this histidine 93 present? It is present on the F helix. A to H is there, right? In which F helix contain the histidine 93, and in the F helix its position is 8th. In the overall polypeptide chain the histidine position is 93, whereas if you take only the F helix its position is 8, hence we call that as F8 also. F8 are proximal histidine, or histidine 93 is the one which connects the heme iron with the globin part. Okay, now this amino acid is covalently bonded to the iron atom. That is what is the 5th coordination. Now what about the 6th coordination? I told you that it is with the oxygen. So iron has 6 coordinate bond. I'll repeat again. 4 with the nitrogen atom of the pyrrole, that is with the porphyrin ring. Then 5th is with the amino acid histidine, and the 6th is with the oxygen atom. That is how it fulfills its 6 coordination bonds. Fine. So here you can see the F helix wherein histidine F8. So here this is a helix with different amino acids. Okay, now here it is the histidine at the 8th position in the F helix, and now this histidine is the one which binds to the heme. Hemali ಯಾವ್ದ್ರ ಜೊತೆ bind ಆಗ್ತಾ ಇದೆ? Again it is with the iron atom. So this is the heme wherein the 5th coordinate bond is with. This is how they are held together, the globin and the porphyrin ring. And with the other, the 6th coordination bond will be with the oxygen. Okay, I think this is again another picture which shows you F helix containing the proximal histidine, or F8 or histidine 93, right? So this is the one, the amino acid which binds to the iron atom of the porphyrin ring. So porphyrin ring il nodi 4 nitrogen irutte, a pyrrole ring idave, 5th one is with here and 6th one is with here oxygen. Okay, So this is again a further detailed structure. This is the histidine. Histidine ನಲ್ಲಿ ಯಾವುದು ಜೊತೆ ಇಷ್ಟೊತ್ತು ಬರೀ histidine ಅಂತ ಹೇಳ್ತಾ ಇದ್ವಿ. Histidine ನಲ್ಲಿ ಯಾವುದು? Here histidine has a imidazole ring, and imidazole ring ನಲ್ಲಿ ಒಂದು nitrogen atom ಇದೆ, and that nitrogen atom is the one which forms a bond with the iron of this heme. Okay, and that is how they are held together. This is the only link, this is the only linkage or the bond formed between the protein part of this hemoglobin and with this prosthetic group. And sixth is with, here you can see, that is with the oxygen atom. Okay, O2. Now coming to the different states in which the hemoglobin exists. One is called as T state and the other one is the R state. And what is this T state? T state is actually the deoxyhemoglobin state. When the hemoglobin is not bound to the oxygen, we call it as deoxyhemoglobin, and this is actually a tense state for the hemoglobin. Whereas R state is the one which is the relaxed state, and this is the oxygenated hemoglobin. Oxyhemoglobin is the R state or the relaxed state, whereas the deoxyhemoglobin is the T state or the tense state. These are the two states in which the hemoglobin exists, are the two forms. Here you can see the deoxygenated state, deoxygenated, or in other words, it is the T state or the tense state. Why it is tense state? Because when this is not bound, when the iron is not bound to the oxygen, the heme is actually little domed in shape, or in other words, it is non-planar. The heme will be non-planar. That is why it is a tensed state. Whereas as soon as the oxygen binds to it, it becomes planar. So before that, it was like a domed. Whereas as soon as it binds, it becomes planar. And in this planar state, it is a relaxed state, we call it as, okay. So the oxygenation makes the hemoglobin relaxed, whereas deoxyhemoglobin is a tensed or the T state it remains. So here, so this is what you can say, the alpha 1, alpha 2, beta 1, beta 2. So as the oxygen binds to, you can make out here the tense state and the, so the binding of one oxygen to one subunit, it makes a change in the entire structure of the hemoglobin. So the binding of one oxygen molecule, it enhances the binding of oxygen to the second subunit, and binding of oxygen to the second subunit further enhances its binding with the third subunit. And similarly, the fourth subunit which also will be stimulated to bind to the oxygen. Okay, so that the hemoglobin now remains in a relaxed state. So the oxy and the, so oxygen stated you can see, and this is the deoxygenated state. It is changing its the shape, you can make out, right? So different vertebrates have a different colors of their blood, right? That is because you know that the humans and the majority of the vertebrates have a hemoglobin which is actually red in color, and some of them also have a blue color. That is because of the presence of hemocyanin. Okay, then green color also can be present in some segmented worms and leeches also, or some marine worms. So that is because of chloro, that is because of the chlorocrurin, which gives it a green color, then violet color because of hemoerythrin. So this is the different forms of the different colors of the blood due to the different pigments that are present there. Okay. Now, to summarize, hemoglobin, as you know, it consists of 574 amino acid residues. It has two alpha subunit, each with 141 amino acid, whereas two beta subunit, each with 146 amino acid residues. And both alpha and beta are held together by ionic and hydrogen bonds, and also by the hydrophobic interactions. Okay, so this is about the structure of hemoglobin. So when you are asked to write the structure of hemoglobin, you have to start with what is the quaternary structure. So what are the number of subunits? Is it a heterooligomer or a homooligomer? Then total number of amino acids, then what is in the N terminal and C terminal of alpha chain and the beta chain. Okay, then it's in its structure, like The tertiary structure you have to explain for the each subunit. Fine, it has an alpha helix structure from A to H, just the same points what you have learned for the myoglobin structure. Okay, so only thing here, myoglobin has single polypeptide chain, that hemoglobin has four polypeptide chains. That is all is the difference. Otherwise, the remaining all structure remains the same. I hope it is clear. If you have any doubt, you can, you can do ask now, or you can clarify. No doubts? Right then. So tomorrow again, same time, 1:30 is my regular class for your section. So we'll meet tomorrow then. So structural organization of protein is over now. So primary structure we have discussed, secondary, then types of secondary, alpha and the beta, then tertiary structure, myoglobin example, then quaternary structure, hemoglobin. So you'll be asked to write, discuss the tertiary structure. They'll ask when you have to take the example of myoglobin only. When you're asked to write, give an account on the quaternary structure or explain quaternary structure taking an example. You have to take this example of hemoglobin and then you have to explain. Okay. So, and for the contact class, have you decided the time? Or I hope your offline classes will start from Friday onwards. I think the notification has to come soon. I mean, if you have read the newspaper or the news in the medias, so after 14, they are going to start the offline class for first year and second year students also, it seems. If that is the thing, how many are interested to attend offline class? Anybody there?
Yes, ma'am.
Yeah. How many of you are interested to attend offline class?
Ma'am, actually, there's one problem. Hostels have not been yet started. College hostel has been started. For the other private hostels are not yet started. So, if they have to come, they have to stay somewhere else. So that will be the problem.
Yeah, that is the problem. So many of you will be opting online only then.
Yes, ma'am.
Okay, right.
Yes, ma'am.
Yeah, I think obviously they have to give you an option also, right? Both offline and on.
Yes, ma'am.
Yeah, so let's
Yes,
see about
ma'am.
that. Okay,
Ma'am.
we'll meet tomorrow then, same time, 1:30. I'll send the link at around 1 o'clock, right?
Oh, okay, ma'am. Thank you,
Super
ma'am.
then. If you have any doubt, you can put your comments in the classroom. I'll try to attend. Fine then, see you.